Hey there, science fans! Today we're diving into the amazing world of cells. Cells are the tiny building blocks of all living things. Inside each cell there's a special molecule called DNA. You can think of DNA as a blueprint. It holds all the instructions for building and running an organism. That's right, everything about you, from the color of your eyes to your height, is determined by the information stored in your DNA. DNA is found inside the nucleus of a cell. Like a captain in a spaceship, this nucleus protects the DNA and keeps it safe. The DNA molecule looks like a long, twisted ladder. We call this shape a double helix. It's like a spiral staircase. Each rung of the ladder is made up of special chemicals called bases. These bases are the letters of the DNA alphabet. There are 4 different bases: adenine, thymine, guanine, and cytosine. These bases pair up in a specific way. Adenine always with thymine and cytosine always with guanine. It's like a matching game. The order of these bases is what determines the instructions in your DNA. Now let's talk about genes. Genes are sections of DNA that contain the instructions for making specific proteins. Proteins are the workhorses of the cell. They do all sorts of important jobs like carrying oxygen, fighting off infections, and helping us digest our food. Think of genes like recipes in a cookbook. Each recipe tells the cell how to make a specific protein. These recipes are written in the language of DNA, using those adenine, thymine, cytosine, and guanine bases we talked about. The sequence of these bases tells the cell what amino acids to use and in what order to build a specific protein. It's like a secret code that tells the cell how to make everything it needs. The amazing thing is that every single cell in your body contains the same DNA. It's like having the entire cookbook in every single one of your cells, but each cell only reads and uses the recipes it needs to do its specific job. So a muscle cell will read the recipes for muscle proteins, while a nerve cell will read the recipes for nerve proteins. Now, how does the cell actually read and use these DNA recipes? That's where RNA comes in. RNA is like a messenger that carries the genetic information from DNA to the protein-making factories in the cell called ribosomes. The process of making RNA from DNA is called transcription. During transcription, the DNA double helix unwinds like unzipping a zipper. Then a special enzyme comes along and creates an RNA copy of one strand of the DNA. Think of it like copying down a recipe from the cookbook onto an index card. This RNA copy, called messenger RNA, is now ready to leave the nucleus and head out to the ribosomes. The messenger RNA carries the genetic code from the DNA in the nucleus to the ribosomes in the cytoplasm, where proteins are made. It's like delivering the recipe card from the kitchen to the chef. Once the mRNA reaches the ribosome, the process of translation begins. Translation is like reading the recipe card and gathering all the right ingredients to make the protein. Remember those bases in RNA? Well, every 3 bases form a code word called a codon. Each codon tells the ribosome which amino acid to add to the growing protein chain. The ribosome reads the mRNA codon by codon, like reading a sentence word by word. Each codon attracts a special molecule called transfer RNA, or tRNA, that carries the matching amino acid. It's like each word in the recipe calls for a specific ingredient. As the ribosome moves along the mRNA, it links the amino acids together in the order specified by the codons, forming a chain of amino acids. This chain then folds into a specific 3-dimensional shape, creating a functional protein. And voila, we have a protein ready to do its job in the cell. This whole process, from DNA to RNA to protein, is called the central dogma of molecular biology. It's like a one-way street of information. DNA holds the master blueprint, RNA carries the instructions, and proteins do the work. Now, there's a little history behind this central dogma. Francis Crick, one of the scientists who discovered the structure of DNA, first proposed it. He said that information flows from DNA to RNA to protein, but not the other way around. It's like saying you can copy a recipe from a cookbook, but you can't put a cake back into the cookbook. James Watson, Crick's partner in discovering DNA's structure, had a slightly different take. He simplified it, suggesting that DNA makes RNA and RNA makes protein. While this is easier to remember, it's not completely accurate. Information can flow from RNA back to DNA in some cases, like with certain viruses. But the main idea of the central dogma still holds true. Genetic information flows from DNA to RNA to protein, directing the formation of all the proteins that make life possible. This process is essential for all living things, from the smallest bacteria to the largest animals. It's the foundation of life as we know it.